Hello everyone, let's get started. In today's video, we're going to be talking about the first question in your laboratory homework, which asks you to put together a graph using the data from our experiment, the inhibition of phagocytosis in Tetrahymena. Now, this data table that you see in front of you is a compilation of data that was collected from several sections of this course across a couple of different semesters. So in each section of the course that ran this experiment, we gathered together all of the data that the students generated and put this data table together so we could do some analysis with it. So let's just recall from the protocol how this table was generated. Remember that the tetrahymena were examined at different time intervals. At the start of the experiment, after 10 minutes of exposure, 20 minutes, 30 minutes. And we had 5 groups in total. We had 2 control groups, the distilled water and ethanol. Those were the 2 solvents. And then we had 3 treatment groups. We had cyclohexamide, colchicine, and cytocollagen B. So for each one of these cells where there is a number, 3 cells were evaluated. 3 tetrahymena cells were evaluated. The ink-filled vacuoles, or the ink-filled phagosomes, were counted up in the 3 different cells, and then a mean was taken, an average was taken. So for example, the mean number of phagosomes at time 0 in the distilled water control group was 0.3, so less than 1 less than 1 phagosome per cell. 0.2 in the other control group, the ethanol control group. Not much better in any of the treatment groups, right? 0.1, 0, 0.1. Now, if you start to go down from there, you can see for the control group, the number of phagosomes that were brought in to the tetrahymena cells steadily increased over the 30-minute period. And compare that to the ethanol, same thing, steady increase in the mean number of phagosomes over the 30-minute period. Now, when we look at the treatment groups, there is one treatment group where we saw that same pattern, and that was the colchicine group. A steady increase in the mean number of vacuoles over the 30-minute period. The cytocollagen B treatment group and the cyclohexamide treatment group look different, though. For both of those groups, we just don't see that increase. We don't see the cells being able to take in more and more and more phagosomes over time. So just from looking at the data, just from eyeballing the data, when we make a comparison between the distilled water and the cyclohexamide, we see a difference we would like to know more about. The distilled water to the colchicine, we don't really see a difference in that pattern. From the from the ethanol to the cytocollagen B, we see a difference. It looks like a steady increase here. It looks like very little, if any, increase here. Okay, so we have some interesting data. We have some interesting data to evaluate. Now, something that you can't see in this view is that we did run some statistical tests, one particular type of statistical test called a student's t-test on this data. And if you recall from your statistics course, the student's t-test compares means. It compares a sample mean, what we actually collect in our physical sample, to the theoretical or the real mean that we would expect for the population if we were able to measure the whole population. And it looks for differences. So the student's t-test was conducted on these three comparisons: the control to cyclohexamide, the control to colchicine, and the control to cytochalasin B, looking for differences, again, between the means in those two groups. And if you recall from statistics class as well, one of the things that we can generate from that kind of a statistical comparison is something called a p-value. Now, p-values are proportions, and like all proportions, you can multiply a p-value by 100 and make it a percentage. Now, the reason that's helpful to us is because when we read a p-value as a percentage, what it says to us is the percentage chance or the percentage probability that this difference that we're seeing between two groups, between a control and a treatment, is due to chance. Let me say that again, because that's really important. When we read a p-value, especially when we read it as a percentage, what we're looking at is a percentage probability that a difference that we're seeing between a control group result and a treatment group result is due purely to chance. It had nothing to do with the treatment. So, for example, let's say you have a p-value of 0.05. That's a very commonly used p-value in research because as a scientific community, sort of as a generalized scientific community, that's sort of the highest p-value that we're going to accept and still think that our difference in our data has statistical significance. So if we have a p-value of 0.05, that's a proportion. We can multiply that by 100 and make it a percentage. 0.05 times 100 is 5%. So remember what the p-value says. There is a 5% probability that any difference we see between the control group and the treatment group is due purely to chance. It has nothing to do with the treatment itself. 5%. Now, remember from statistics class that if you have something that has a probability of 5%, then the opposite of that has a probability of 100 minus 5%, or 95%. So if there's a 5% probability that the difference we're seeing is due to chance, then there's a 95% probability that the difference that we're seeing is real. It's due to the treatment itself. So we're going to use 0.05 as our cutoff for statistical significance. We want to see a p-value of 0.05 or less for us to conclude that any differences we're seeing are statistically significant. All right, let's look at some p-values. Okay, down at the bottom of the screen, you can see that when we compared the control to colchicine, the p-value came in at 0.20. When we compared the control to cyclohexamide, the p-value came in at 0.03. And when we compared the control to cytochalasin B, the p-value came in at 0.05. Now, excuse me while I fix a spelling error right here. It's actually cycloheximide. All right, so let's first convert these p-values to percentages. 0.2 is the same as 20%. 0.05 0.03 is the same as 3%, and 0.05 is the same as 5%. Now, I'm going to use the rule that says if there's a probability of something happening, then the opposite probability is 100 minus that probability. So 100 minus 20% is 80%. 100 minus 3% is 97%. 100 minus 5% is 95%. This column is what's the probability that any differences that we saw were due purely to chance. So I'm going to label that as chance. And this column tells us the probability that any differences we saw were due to the treatment. Let's take a look at these results. When we treated the Tetrahymena cells with colchicine, the p-value tells us that any differences that we saw between the control group and the colchicine group, there was a 20% probability that those differences were due purely to chance. That means there's an 80% probability that those differences were statistically significant, that those differences were due to the treatment itself. Now, remember, we looked at the graph of just the colchicine versus control when we were together in lab. Let's pull that graph up. Okay, I've pulled that graph in on top of our data table here. Remember what you're looking at? Across the x-axis, this is time going by. Across the 30 minutes of our experiment. Across the y-axis, this is the mean number of ink-filled phagosomes that we saw in the Tetrahymena. The blue line represents the colchicine treatment group. The orange line represents the control. Now remember, when we evaluate at 30 minutes, when we compare what these lines look like at 30 minutes, there's a difference here. It's not exactly a difference that you would expect, because it looks like colchicine may have made it easier for these cells. Exposure to colchicine maybe made it easier to perform phagocytosis. It didn't inhibit it. It doesn't look like it did anyway. So maybe there's a difference here. Maybe there's a small difference here. But when we tested that, we got this p-value. And the p-value tells us that that difference that we see at 30 minutes, there's a 20% probability that it's not real. It only happened by chance. And if we repeated the experiment again and again and again and again, maybe next time we wouldn't see this difference, or maybe the difference would be swapped, or in other words, it's not real. It's not a real difference. It's not a repeatable difference. It's not a difference that was really due to the effects of colchicine. It was simply chance that created that difference. Now, let's look at the next comparison that we made. When we compared the control to the cyclohexamide inhibitor, we got a much lower p-value, didn't we? We got a p-value of 0.03, 3%. So any difference we saw between the control and the cyclohexamide, any difference, there was a 3% chance, 3% probability that that difference was just a fluke, but a 97% probability that it was real, that it was actually due to the cyclohexamide. Let's pull that graph up. Okay, here's that graph. Again, time on the x-axis, mean number of ink-filled phagosomes on the y-axis. The blue line is the control. The orange line is the treatment. When we look at this graph, we're struck by the difference, right? I mean, it strikes you visually that while the control group just ate and ate and ate phagosomes, the treatment group, it looks like it just flatlined after about one phagosome coming in. It just flatlined. It looks like there's a big effect. It looks like exposure to cyclohexamide inhibited phagocytosis from occurring. When I look at the p-value, that is confirmed. Remember, we said we're going to make a cutoff for statistical significance at 0.05 or 5%. I'm below that. I'm at 3%. There's a 3% probability that this effect that I'm seeing, this difference that I'm seeing, isn't real. It's just a fluke. And that if I repeated the experiment a bunch of times, it would go away. There's a 3% probability of that. That means there's a 97% probability that this is real, that the cyclohexamide group really did have a significant drop in mean phagosome number. There's a 97% probability that the difference I'm seeing here is due to the treatment. It's due to the cyclohexamide, that the cyclohexamide really did inhibit phagocytosis. In other words, that this is a statistically significant result. Finally, we have ethanol versus cytocollagen B with a slightly higher p-value, but still within our limits. P came in at 0.05. That's a 5% probability that any difference was due to chance. Let's pull up that graph next. Here's the graph comparing the control in blue to the treatment, the cytocollagen B, in orange. It looks similar to the last graph, doesn't it? At 30 minutes, which is again where we're evaluating our data, there appears to be a very big difference between the mean number of phagosomes in the control group and the mean number of phagosomes in the treatment group. It looks like the treatment group has many fewer phagosomes. That's what this data sure looks like. It looks like the cytocollagen B had an effect. It inhibited phagocytosis, at least to a large degree. When we go to the p-value, that's confirmed. The p-value tells us there's a 5% probability that this difference that I can see with my eyes isn't real. It's just due to chance. But there's a 95% probability that this is real. That the difference I'm seeing here was due to the cytocollagen B itself. That the cytocollagen B actually did inhibit phagocytosis to this degree. So I have 3 p-values. 2 of those p-values tell me that my treatment was statistically significant in reducing the ability of those Tetrahymena to perform phagocytosis. 2 of these p-values tell me that the drop I saw in the mean number of phagosomes in the treated cells, that drop is a statistically significant difference. I have 1 result that tells me there is no difference. There is no statistically significant difference between the control group and the colchicine group. So the colchicine did not inhibit phagocytosis in my Tetrahymena cells. All right. Next, we're going to take a look at how we're going to graph these results, because we're going to create one graph that has all of our groups on it. 2 control groups, 3 treatment groups. And we're going to create that graph in Excel. Okay, so we're back to our data table, and you'll see below the data table that there are some instructions for how to make your graph in Excel. The very first thing you have to do is tell Excel which data from the table you want to include in the graph. So all you have to do is click and then drag and include all of this material. This tells Excel that these are the variables and the data points that you want to include in your graph. Now remember, Excel refers to a graph as a chart. So I have to go in and ask Excel to insert a chart. So if you go up to the menu bar, you can click where it says Insert. I'm also going to come down here and I'm going to re-highlight my data because I got that removed. Now, in my version of Excel, and your version may be slightly different, but in my version, I can go up where it says Recommended Charts and click, and I can scroll down through all of these charts until I see the one that I want. And of course, this is the one that I want right here. I'm going to click on that, and there's the skeleton, at least, of my graph. Now you'll see in the instructions down here, it says insert the chart called XY scatter. And of course, this was referred to as a line graph, not an XY scatter. So I'm going to right click on this, and I'm going to scroll to where it says change chart type. And from there, I'm going to click on XY scatter. Now, there are lots of different versions of the XY scatter chart here. You'll see that I'm offered just a line. Hold on just a sec. I'm offered a version that shows just a smooth line. I'm offered a version that shows a smooth line with some points on it. I have just points. I have points with a staggered line, and I have just a staggered line. Now, for our graph, we're going to choose the smooth lines with the points as our XY scatter. So I'm going to go ahead and click on that, and it has updated my chart to show that. Now, if we scroll down in the directions, you'll find that there are a couple other things that you have to do to get your chart ready for submission, get your graph ready. You're asked to delete the chart title. You're asked to check the features of your legend. You're asked to put a title on your x-axis and put a title on your y-axis. So we have a couple other things that we need to do to get this graph ready for submission. I want to say just a couple words about chart titles. When we make a graph, we're making it either for presentation to a living audience, or we're making it for publication in a written document. By convention, if you're going to put a title on a graph, you are preparing to present that graph to a living audience. So either you're doing a presentation in a lecture hall to a group of people, or maybe you're at a poster conference where people are walking past your poster and you're presenting to a living person. If that's the case, it's appropriate to put a title on your chart. If you're going to be presenting this graph in a written publication, let's say you're preparing to make a written report, or you're preparing to send a manuscript to a journal for publication. In that case, we don't use a title. Instead, we use what's called a caption underneath the graph. So one of the things that you're asked to do in this assignment is get rid of this. And you can see all you have to do is just delete it. Excel shows you a place where you can add your title. If you don't need your title, you just delete it. Now, you're not actually going to put your caption into your graph in Excel. Instead, once your graph is all finished, you're going to copy and paste it into the Word document that is your laboratory homework, and you're going to type your caption underneath your graph in Word. So we're not going to deal with the caption just yet, but I do want to say for a minute what belongs in a caption. A caption is a description for the reader, because again, this would be what you would put into a written document. A caption is a description for your reader of what the graph shows them. So we always begin a caption by saying something like figure 1 or table 2. We give it a descriptive name, and then we write out in written form what it is that they can see in this object. That's all a caption is. It's nothing to get, overwhelmed with when you're writing it. So we have to get rid of the chart title. When we're ready, we're going to write a caption for this graph, but we also have to do a couple of other things. We need to make sure we have a descriptive legend, which we do because Excel has already put that in for us. And we also have to make sure that we have descriptive labels on both of our axes. So we're going to have to go back up into our menu bar here and make labels for our axes. The first thing you have to do is click on your chart. Remember, when you click on a graph in Excel, you're telling Excel to pay attention to it. I want to do something with this graph. Then you're going to want to go up into your menu bar, and you're going to want to click where it says Chart Design. If you click on that, way over on the left-hand side, there's something that says Add Chart Element. Go ahead and click on that arrow to see your down menu. And in here, you can put in all different kinds of things. You could add a chart title if you wanted to. You could label data. You could add error bars. You can add trend lines, all different kinds of things. But we just want to title our axes. So I'm going to start with the horizontal axis or the x-axis. You can see Excel has added basically a text box for me. Now remember, the most critical thing about an axis title is the units. Tell your reader what you're measuring this thing in. So our x-axis reflects time passing by, and we measured our time in minutes. Now I'm going to go back up to chart design, back over to add chart element, and I'm going to add a primary vertical. Click on that. And remember, our y variable is something very specific. It's the mean number of ink-filled phagocytomes in our Tetrahymena. Okay, I have a little spelling error there I can fix. The mean number of ink-filled phagocytomes that we found for each time point and each grouping. So now I have my graph ready to go. My graph shows for my reader 5 individual lines in different colors, representing the 2 control groups and the 3 treatment groups. Now, recall from the instructions that you're not going to submit your graph in Excel. You're going to highlight your graph, then you're going to right-click and copy it, because you're going to paste it into a Word document. Here I have opened up the assignment sheet in Word. I'm going to scroll down, and I'm going to paste my graph right here after question number 1. I'm just going to right-click and ask to paste. Here is the pasted graph. I'm just going to tweak it a little bit so it's a size that looks reasonable to me. Now, don't forget, you're not quite done because you have to add a caption here. The cursor almost always ends up on the lower right-hand side of the object that you're cutting and pasting. So I'm going to go ahead and enter, and then I'm going to enter again, and I'm going to write my caption. So for a graph, we call it a figure. So since this is the first figure that I've put into this document, I'm going to call it figure 1. Now I have the job of explaining to the reader what they can see in front of them. So I'm going to tell my reader what this is. This graph shows the mean number of ink-filled phagocytomes in Tetrahymena piriformis exposed to either cyclohexamide, colchicine, or cytocollagen B over a 30-minute period. There, I've written a sentence that summarizes for the reader what they're looking at. Now, you don't want to end your caption here. You want to add a little bit more for your reader, because the other thing that your reader can see is that some of those lines look very similar and some of those lines look very different. So you want to add another sentence or two describing in words where those differences are and are not. Example, you could write something to the effect of, when T. piriformis was exposed to colchicine, there was no statistically significant difference in mean number of ink-filled vacuoles compared to the control. And tell your audience, tell your reader what that control was. In comparison, exposure to both cyclohexamide and cytocollagen B resulted in a statistically significant decrease in the mean number of ink-filled vacuoles, or phagocytomes, I'll say, in the cells when compared to the controls. Now, I wrote cyclohexamide and cytocollagen B, so I'm going to say DiH2O and ethanol, respectively. That tells my reader the DiH2O is for cyclohexamide, the ethanol is for cytocollagen B. Now, is this exactly how you have to write a caption? No. You put a caption in your own words. You could word this a couple of different ways. You could say, exposure to colchicine produced no significant change in mean number of ink-filled phagocytomes compared to the control. you could say, you could begin with the other two treatments. You could say there was a statistically significant difference or a statistically significant decrease in the mean number of phagosomes in T. piriformis exposed to cyclohexamide. And then you could write something, I should say, compared to its control. And then you could write something different, something separate for the cytochalasin B. There's all different ways you can state what the graph shows. The important thing is that you want to tell the reader what you know about your comparisons, that there's no difference between the distilled water and the colchicine, that there is a difference between the distilled water and the cyclohexamide, and there is a difference between the ethanol and the cytochalasin B. Statistically significant differences for these two treatments, no difference for this treatment. You could also include the p-values in your caption. You could tell the reader what your p-values were for those three comparisons. There are all different ways that you can tell the reader what exactly this particular graph is showing them in terms of your results. So, do your best when you're writing your caption to be thorough and tell your reader what they're looking at. Okay, so that brings us to the end of this video. Remember, if you're using a different version of Excel than this one that I've shown you today, you may have to look up a tutorial for yourself on YouTube in order to find each of the different features that we went through today in terms of putting together an XY scatter graph and adjusting all of the labels. But you should be able to find one without any difficulty.